Didarul, instructor of BIO201, Introductory to Biochemistry and Biotechnology. So today we'll study class 2, lecture 2, its foundations of biochemistry. So today we will study foundation of biochemistry. So all the animals, plants and microorganisms and human have the same basic structural unit, and this is cell, and the same kinds of macromolecules like DNA, RNA and proteins. So biochemistry is the field of the chemistry of living organism, and there are 5 foundations of biochemistry. One is cellular, another is chemical, another is physical or thermodynamic, and fourth is genetic, and fifth is evolutionary. So let's start with the cellular. Okay, so all the cell have plasma membrane and DNA and cytoplasm, and the 3 major components of the cell. So the nucleus in eukaryotics or nucleus in prokaryotics that contains the DNA, and the plasma membrane that surrounds the cell, and cytoplasm that contains the organelles. So the cell that is nuclear envelope, the cell with nuclear envelopes are called eukaryotic, and the cell where the nuclear or genetic materials are not covered or not surrounded by nuclear membrane is called prokaryotics. Okay, and here are the contents of nucleus in eukaryotics and bacteria. So the nucleus that contains genetic materials, DNA and associated proteins, and nucleus is membrane bounded in eukaryotic cell, and nucleus does not contain any membrane in bacteria, and that is called nucleus. And this is the plasma membrane, and this is the top flexible lipid bilayer and selectively permeable to polar substance, and this includes membrane proteins also that functions in transport, signal receptions, and as well as an enzyme. And in the cytoplasm, this is an aqueous cell contained and suspended particular and organelles. So, and this is the eukaryotic cell, and this is the prokaryotic cell. And when you centrifuge the cell lysate or cell in centrifuge machines, so you will get 2 parts. One is supernatant and another is pellet, and the supernatant contains cytosol, and this is a concentrated solution of enzyme, RNAs, monomeric subunits, metabolites, and inorganic ions. And pellets contain particles and organelles like ribosomes, stores granules, mitochondria, chloroplasts, lysosomes, and endoplasmic reticulum. So there are common and distinct features between animals and plant cell. So what are the common structure in both animals and plant cell? So plasma membrane, nucleus, ribosomes, peroxisomes that contains digestive enzymes for breaking down the toxic materials, and the cytoskeleton, Golgi complex, smooth endoplasmic reticulum, rough endoplasmic reticulum. And there the animal cell specific structure is lysosome, and there are several plant cell specific structure like cell wall that is absent in animal cell, vacuole, chloroplast, plasmodesmata, and glioxysome. So plasmodesmata that connects to plant cell through a link between a cell wall, and glioxysome that is the cellular organelles that converts lipids to sugar during the early stages of germination in oil seed. So cell have 4 levels of molecular organization. One is monomeric unit, another is macromolecules, another third is supramolecular complex and cellular organisms. So here is the example. So level 1 monomeric units like nucleotides work as a monomeric unit of DNA, and this is the macromolecules. So level 2, and amino acids are monomeric units, and the macromolecules are protein, and sugars are the monomeric unit. The macromolecules are cellulose, glycogen, etc. And the macromolecules then form supramolecular complexes, so like DNA to chromosome, protein to plasma membrane, and cellulose to cell wall. And then the final level, level 4, the cellular and its organelles. Okay. So cell builds macromolecular structure. Like amino acids are organic compounds that combine together to construct proteins, and this is the building blocks, and this is the macromolecular structure. And sugars like glucose, fructose, galactose combine to construct carbohydrate like glycogen, starch, and nucleotides combine together to form nucleic acid like DNA and RNAs. So let's go to the chemical foundations or chemical basis of biochemistry. So here is the element required for living organism. The bulk element that is present in abundant amount is hydrogen, carbon, nitrogen, oxygen, sodium, phosphorus, sulfur, chlorine, potassium, and calcium. And the trace elements like micro elements, also called minerals, like manganese, chromium, manganese, magnesiums, irons, okay, cobalt, nickels, okay. So here is the basic information about biomolecules are based on carbon atoms. So carbon is the fourth most abundant element in the universe, and carbon is a highly reactive element. So carbon has 4 valence electron that can form 4 bonds with other atoms. So 1, 2, 3, 4. As you know, this is a very fundamental study, and carbon basically makes bond with hydrogen and oxygen and nitrogen. So hydrogen has 1 valence electron, oxygen has 2 valence electron, nitrogen has 3 valence electron. So here is the methane. So 1 carbon can bind 4 hydrogen, and 1 carbon can bind 2 oxygen atom, making carbon dioxide. And here is the alanine. So 4 different group of atoms bind with the 1 carbon molecule. So here are some common features or some common functional groups of biomolecules. So what is functional groups? You know, you have already studied about the functional group. Functional group is an atom or group of atom in an organic compound that defines the structure of a family of compounds and determines the properties of family. So here are some common functional groups of biomolecules, like methyl group, ethyl group, phenyl group, sulfahydryl group, that is found in the cysteine, an amino acid, and disulfide bond. This is disulfide group found in the peptide or protein, and carbonyl group found in the carbohydrate like aldehyde, and ketone found in the carbohydrate. Carboxyl group found in the amino acid. Hydroxyl group found in the glycerol, and phosphoryl group. This is also found in the ATPs, or amino group NH2, and amide group CONH. Okay. So let's study the physical or thermodynamic basis. So energies for living organism. Energies for living organism, that means all living cells and organisms must perform work to stay alive or to reproduce themselves. And all anabolic or catabolic or synthetic reactions in the cell require energy, and nearly all living organisms derive their energy from sunlight. So to study the thermodynamics of a cell, we need to know what is entropy and what is enthalpy. So entropy, this is the randomness or disorderness of the components of a chemical system, is called entropy, and expressed is as. So any changes in the randomness of a system is expressed as entropy changes. So what is enthalpy is denoted by H. This is the total heat content of a system. Let's say we can present this schematically here. So glucose and 1 molecule of glucose react with 6 molecules of oxygen. That means the atoms contained in 1 molecule of glucose plus 6 molecules of oxygen, a total 7 molecules, are more randomly dispersed by the oxidation reaction and are now present in a total 12 molecules. That means 1 molecule of glucose and 6 molecules of oxygen, total 7, and 6 molecules of carbon dioxide and 6 molecules of water, total 12. So 7 molecules converted into 12 molecules. So this is the increasing of randomness. Look at here, 7 molecules and 12 molecules. So this is the increasing of randomness. Okay, and also this is the gas, and is also the randomness, increasing the randomness of reaction. So whenever a chemical reaction results in increasing the number of molecules, or when a solid substance is converted to liquid or gaseous products, which allows more freedom of molecular movement than solid, and molecular disorder, and thus the entropy is increased. So Gibbs free energy and enthalpy. We know Gibbs free energy is the energy that is available to do useful work. So Gibbs free energy is called free energy because Gibbs energy is easily available anytime. It requires the reaction can get this energy without having to work for it. So free energy content denoted by G can be defined in terms of 3 quantities like enthalpy, entropy, and temperature. So when a chemical reaction occurs at constant temperature, the free energy changes delta G is determined by the enthalpy change and the entropy changes. So free energy change delta G is equal to delta H minus delta S. while temperature is constant. So changes in Gibbs free energy is equal to total free energy changes of a system minus free energy lost in disordering the system. So if the reaction is endothermic, that is delta H is negative, then the entropy is increased, and this is delta entropy is positive, randomness is increased, and delta G must be negative and reaction can be processed. So if the reaction is endothermic, that means delta H is positive, then the entropy decrease, delta S is negative, then delta G is positive, that means reaction cannot be processed. So for a favorable condition, delta G always be negative. So what is free energy change? Actually, entropy of a whole universe can never be decreased. So it can never be negative, and in any isolated system, entropy always increases. Okay, that means things are getting more disordered. Look at here. So enthalpy and entropy changes. So this is the enthalpy change and this is the reaction proceed. So this is the reactant, the energy content of the reactant, and this is the energy content of the product. So energy content of the reactant is higher. When this reactant converted to product, the energy will be released, and this is the free energy and this is the enthalpy changes. So if the free energy is negative for reaction, then this is the favorable reaction. So delta H is negative. As the reaction proceeds, the enthalpy is going down, and enthalpy is converted into heat and release. Okay, so here delta H is negative because this minus this, energy content of product minus energy content of the reactant. So this, the delta G is negative here. So let's study the activation energy. So all the chemical reactions need an initial investment of energy is called activation energy to go to the transition state. So activation energy required to begin the reaction state. Okay, so this is the activation energy. So what is exergonic and endergonic reactions? A chemical reactions in which the free energy changes is positive and energy is absorbed, and this is endergonic reaction. So when the energy content of reactant is lower than the energy content of the product, the energy will be absorbed, and this is called endothermic reactions. So the example is photosynthesis, and when the energy content of the reactant is higher than the energy content of the product, then energy will be released, and this is the delta G is negative and this is the delta G is positive. So this is the exergonic reaction. So exergonic reaction means energy will be released, and endergonic reaction, energy will be absorbed. Exergonic reaction means delta G will be negative, endergonic reaction is delta G is positive. So coupling of endogenous and exogenous reactions in biology. So here, let's say glucose is energy contained, energy rich nutrient. When it's break down to carbon dioxide, water, and heat, this is the energy releasing process. That means exergonic reaction. That means breakdown of glucose to those compound releases energy. And in the thermodynamics, there is a law that means energy cannot be synthesized and created. So when energy is released from glucose, there should be something who can accept that energy. So that here ADP react with PI phosphate to form ATP. So here the bond will be stored here. So this is the endergonic reactions, and this is the exergonic reaction. That means breakdown of glucose to heat is the exergonic reactions, and absorption of energy is called endergonic reaction. So ATP synthesis is an endergonic reaction, and when ATP is breakdown to ADP and PI, this is also ATP breakdown is exergonic reactions, and formation of amino acid to protein is called endergonic reaction. That means bond breaking is exergonic and bond making is endergonic. Okay, so let's study the foundations of biochemistry in genetic basis. So this is the genetic information. So someone says, can you show the DNA and RNA visually? The student shows like that. So this is the RNA single stranded, and this is the DNA double stranded. So we know deoxyribonucleic acid or DNA is an organic chemicals that contains genetic information and instruction for protein synthesis, and ribonucleic acid is an important biological macromolecules that functions to convert the genetic informations of DNA to protein. So DNA is a molecule that can store genetic information for longer period of time, and RNA that store the genetic information for short period of time, and it transfer the genetic information from DNA to a protein. Okay, so DNA stores all the chemical reactions. Here is the central dogma. That means DNA to DNA is replication, DNA to RNA is transcription, RNA to DNA is reverse transcription, and RNA to protein is a translation. This is the central dogma. And here the nucleotides is monomeric unit of nucleic acid and form DNA, and DNA is supercoiled here, and coiling of supercoil is organized into chromosome and then is organized into nucleic acid. Okay, so how DNA is converted is information to RNA to protein. So let's say this is the gene, this is the segment of DNA, and it contains the genetic information. So it let's say here is 3 gene. The first gene can produce first RNA, second gene can produce RNA 2, and third gene can produce RNA 3. So translation of RNA sequence into protein sequence and folding of protein in negative conformation. So then it will produce protein 1, it will produce protein 2, it will produce protein 3. Then 3 subunit will link together to form a protein, and this is the production of the gene. That means this is the product of gene. Okay, so let's study the foundation of biochemistry in the evolutionary basis. So here biological evolution on Earth began more than 3.5 billion years ago. So it's too far. And evolution is the process by which different kinds of living organisms are thought to have developed from earlier forms during the history of life. So this is the Big Bang. From there nothing, from there everything has been produced from nothing, and here it occurs in 14 billion years ago, and the human being that developed in 1.8 million years ago. Okay, so heritable changes in DNA drive evolution. Mutation is defined as random change in DNA of an organism that result in a new characteristic that can be inherited. So for example, for mutation, there are 2 differences in DNA sequence in FOXP gene between human and chimpanzee. So chimpanzees are thought to be the more closer ancestor of human being. So the only difference is the FOXP2 gene, and this is called a speech gene, and it is required for proper development of speech and language. So this gene has been properly developed in human being but not developed in chimpanzee, so they are not able to talk and we are able to talk. Okay, so that's all from the class 2 lecture 2, and thank you very much, and we'll next we will start class 3 next time. Till then, goodbye.